In today's video we are going to talk about mushroom cultivation. The term mushroom means in general a fungus, but commonly it is the fruiting body of some fungi which produce and disseminate spores. Like all other fungi, they lack chlorophyll and thus cannot produce their own food. They grow saprophytically or sometimes symbiotically upon other dead and living plants respectively to obtain organic matter as food. Mushrooms are variable in size and shape. Many have cap and stalk, but some varieties are devoid of stalk. Some varieties even produce fruit bodies below the ground. There are a large number of species growing wild in nature, while many are edible, some are highly poisonous. The cultivation of mushrooms is known as fungiculture. In India, they have started to cultivate mushrooms from 1943. Types of mushrooms. Edible mushrooms are fungi that are safe for human consumption and do not cause any harmful effects when eaten. Examples are Agaricus bisporus, Pleurotus ostritus, Lentinula edota, and Hericium arenaceus. Non-edible or poisonous mushrooms, on the other hand, are fungi that contain toxins that can cause serious harm or even death when ingested. Amanita phalloides, Gyromitra esculenta, Galerina marginata, and Conicide phalaris are examples of non-edible or poisonous mushrooms. Life cycle of a mushroom. There are 4 basic stages to the life cycle of a mushroom: spore germination, colonization, fruiting, and sporulation. Of the many spores produced by a mushroom, only a tiny number will land where they can germinate to produce hyphae. Hyphae of different species may grow in soil or in wood and may later form a tiny mushroom button. This then grows, and the stalk and cap expand to become a mushroom. Economic importance of mushrooms. Food. Mushrooms are edible and are considered a delicacy in many cuisines around the world. They are a source of protein, vitamins, and minerals, and have been used for both culinary and medicinal purposes for centuries. Agriculture. Mushroom cultivation is a rapidly growing industry and is becoming an important source of income for many farmers, especially in developing countries. It is relatively low cost and low tech, and can be done in small scale and semi-commercial operations. Biotechnology. The enzymes, antibiotics, and other compounds produced by mushrooms are used in a wide variety of products, including medicine, food, and industrial materials. Environmental benefits. Mushroom cultivation can help to reduce waste and pollution by using agricultural and industrial waste as a substrate for growing mushrooms. This can help to reduce the amount of waste that ends up in landfills, and also helps to reduce the need for the use of pesticides and chemical fertilizers. Bioremediation. Some mushroom species can be used to clean up contaminated soil, air, and water by breaking down pollutants and heavy metals. They are known as mycoremediators. Nutritional and medicinal aspects of mushrooms. Mushrooms are popular for their delicacy and flavor. They are excellent sources of vitamins, proteins, and minerals. They are good source of vitamin B, folic acid, the blood-building vitamin, useful in anemic condition. They also contain pantothenic acid, vitamin B12, ascorbic acid, and the precursor of vitamin A and D. They are good source of phosphorus, potassium, iron, copper, etc. Mushrooms contain all essential amino acids, particularly L-lysine and L-tryptophan. Mushrooms contain least quantity of carbohydrates as well as fats. Mushroom cultivation techniques. Indoor cultivation, in which mushrooms are grown in a controlled environment, such as a greenhouse, grow room, or laboratory. Outdoor cultivation, where mushrooms are grown in natural or semi-natural environments, such as on logs, stumps, or beds of straw or compost. Agar culture is a technique used to grow mushrooms on a nutrient-rich medium made of agar and other ingredients. In liquid culture, mushrooms are grown in a liquid medium, such as a nutrient broth or sugar water. Requirements for mushroom cultivation. Substrate. It is the material on which the mushrooms grow, such as sawdust, straw, or compost. The substrate must be nutrient-rich, free of contaminants, and prepared to the correct moisture level and pH for the specific mushroom species being grown. Spawn. It is the mycelium of the mushroom that is used to inoculate the substrate. It can be purchased or produced from a culture of the mushroom mycelium. Temperature. Different species of mushrooms require different temperature ranges for optimal growth. Generally, the temperature should be kept between 18 to 24 degrees Celsius during the spawn run and 12 to 18 degrees Celsius during fruiting. Humidity. Mushrooms require a humid environment to grow. The humidity level should be kept between 80 to 90% during the spawn run and 85 to 95% during fruiting. Light. Some mushroom species require light for proper growth and development, while others can grow in darkness. If light is needed, it should be provided at the appropriate intensity and duration for the specific species being grown. Ventilation. Proper ventilation is necessary to maintain the correct temperature, humidity, and CO2 levels. Good air exchange can also help prevent the buildup of contaminants and improve mushroom growth. Sterilization. Contamination by bacteria or other fungi can be a major problem in mushroom cultivation. Proper sterilization of the substrate, equipment, and working environment is essential to prevent contamination and ensure successful mushroom production. General steps in mushroom cultivation. The general steps involved in mushroom cultivation are strain selection, substrate preparation, spawning, casing, incubation, fruiting, harvesting, and canning. Strain selection. The first step in mushroom cultivation is to select the strain of mushroom that you want to grow. This is an important decision, as different strains of mushrooms have different characteristics and requirements for growth. Factors to consider when selecting a strain include the intended market, yield, flavor, texture, and disease resistance. Substrate preparation. Once you have selected a strain, the next step is to prepare the substrate. The substrate is the material that the mushroom mycelium will grow on. Common substrates include straw, sawdust, and compost. The substrate must be sterilized or pasteurized to eliminate competing bacteria and fungi. This is typically done by heating the substrate to a specific temperature for a set amount of time. These are the images showing the preparation of a substrate. Steps in preparation of substrates. The steps for preparing substrates for mushroom cultivation may vary depending on the type of substrate being used and the mushroom species being grown, but the general steps are obtaining the substrate, pre-treating the substrate, adding supplements, adjusting pH, sterilizing or pasteurizing the substrate, and finally, cooling the substrate. Spawning. Spawning is the process of introducing mushroom mycelium into a substrate, which provides a nutrient-rich environment for the mycelium to grow and colonize. Mycelium is the vegetative part of a fungus, and it consists of a network of thread-like structures called hyphae. There are different methods of spawning, depending on the type of mushroom and the substrate being used. Some common methods include spore syringe. A syringe containing mushroom spores is used to inject the spores into the substrate. Liquid culture. A liquid culture of mushroom mycelium is mixed with the substrate. Grain spawn, in which the mycelium grown on grains such as rye or wheat is mixed with the substrate. Sawdust spawn, which is mycelium grown on sawdust, is mixed with the substrate. Casing. Casing is one of the steps in mushroom cultivation. Casing involves the covering of compost with a thin layer of sterile soil or materials like chalk powder to maintain moisture and gaseous exchange. The material used to cover the compost must be neutral or slightly alkaline. Casing of mushroom beds or spawn run compost is necessary because casing soil is a nutrient-deficient medium, which helps in converting the vegetative phase into fruiting. Fruit bodies are formed in abundance and thus production is economical. Incubation. Incubation is the stage in mushroom cultivation when the inoculated substrate is kept in a warm, humid environment to allow the mycelium to colonize the substrate. Choose an incubation location. The location for incubating the mushroom substrate should be clean and free from contaminants. A clean room, closet, or greenhouse can be used, and the area should be well ventilated to prevent a buildup of carbon dioxide. Maintain proper temperature and humidity. The ideal temperature and humidity for incubating mushroom substrate depend on the species being grown, but most mushrooms prefer temperatures between 18 degrees Celsius to 27 degrees Celsius, and humidity levels between 80% to 95%. A humidifier or mister can be used to increase the humidity in the incubation area. The mycelium will begin to colonize the substrate, forming a network of white, thread-like strands called mycelial growth. The growth should be monitored for signs of contamination or irregular growth. Any signs of contamination should be addressed immediately. Fruiting. After the mycelium has colonized the substrate, it is time to initiate fruiting. This involves creating the right conditions for the mushrooms to grow. This typically involves exposing the substrate to light and lowering the temperature slightly. The exact conditions required will depend on the strain of mushroom you are growing. Harvesting. Once the mushrooms have reached the desired size and maturity, they are ready to be harvested. To harvest the mushrooms, grasp the stem near the base and twist gently to detach the mushroom from the substrate. Alternatively, a sharp knife can be used to cut the stem near the base. It is important to avoid damaging the substrate or other mushrooms in the process. Canning. Mushrooms can be canned for long-term storage. This involves packing cleaned and sliced mushrooms into jars and processing them in a pressure canner. Steps involved in canning process include: pre-cleaning, mushrooms are cleaned to remove foreign particles, soil, etc. Washing, mushrooms are washed in water. Blanching, mushrooms are blanched in hot water having 0.2% citric acid for 3 to 5 minutes. This process results in loss of 30% weight. Cooling, blanched mushrooms are cooled through continuous counterflow cooling system. Grating, mushrooms are grated according to size. Slicing, mushrooms are sliced for the required size. Filling, the cans are filled and weighed. Brining and exhausting, hot brine solution is added and temperature is raised to 80 degrees Celsius in the center of can to exhaust. Concealing, cans are sealed with lid. Retorting, the sealed cans are sterilized at 15 psi for 15 to 20 minutes. Labeling and packing, the cans are labeled and packed in cartons. Pests and diseases of mushrooms. Pests. Different insects and nematodes grow on mushroom bed and will be responsible for yield loss. Even rats eat away fruit bodies along with grains and other things causing yield loss. Springtails and sciarid flies are common examples for insects. Nematodes, Dactylenthus myceliophagus, affect mycelial growth. Rats damage the beds and eat grain spawn. Control measures of pests. Maintain cleanliness. Prevent compost and straw from coming in contact with soil. Treat the tools with 2% formalin. Spray dichlorovas. Spray Biocuelearning to control mites. Baiting and killing of rats. Diseases, different parasites grow on growing mushrooms in beds, cause characteristic diseases and responsible for considerable loss to fruit bodies harvest. Fungal diseases includes dry bubble, wet bubble, etc. Bacterial diseases are bacterial pit and bacterial brown blotch. Viral diseases are elongated bend stipes and disintegration of mycelium. Weed molds and competitors. The following molds grow on growing mushrooms and will be responsible for yield loss: false truffles, white plaster mold, and ink cap. Mushrooms cultivated in India. Button mushroom, Agaricus bisporus button mushrooms are the most widely cultivated mushroom in India and are grown commercially in many states. They are typically grown on composted substrates made from wheat straw, poultry manure, and gypsum. Oyster mushroom, Pleurotus species, oyster mushrooms are also widely cultivated in India, especially in the northeastern states. They are typically grown on a substrate of rice straw or a combination of rice straw and other agricultural waste products. Milky mushroom, Calocybe indica, milky mushrooms are grown in India and are known for their high nutritional value. They are typically grown on a substrate of paddy straw or a mixture of paddy straw and other materials such as wheat bran. Shiitake mushroom, Lentinula edota. Shiitake mushrooms are grown in India, primarily in the Himalayan region. They are typically grown on a substrate of sawdust or a mixture of sawdust and other materials such as rice straw or wheat bran. Future of mushroom cultivation, increasing awareness of nutritive and medicinal value of mushroom. A boost in cultivation of mushrooms resulted in mass scale. Cultivation of mushrooms helps to convert agro-wastes into human food. Their cultivation provides labor employment as they are fast-growing and are responsible for production of quality food. Substrate/compost preparation with special reference to fermentation, collection of strains of mushrooms from different geographical regions and their evaluation use in breeding work. Improvements in prolongation of shelf life and canning and processing will also boost the prospects of mushroom cultivation.